Okay, let's finish this chapter off. So last time we went over the Kelvin cycle. We also went over the alternative pathway of photorespiration when we didn't have CO2 readily available to us, and our lovely Rubisco enzyme, instead of fixing carbon dioxide, fixed O2 to combine with our Rubisco. So now we're going to go over two other alternative pathways. First one is going to be with C4 plants. Okay, so different types of plants here. These are things like corn and sugarcane and crabgrass. So the nice thing about C4 plants is they have actually evolved past photorespiration. So they're not doing photorespiration if carbon dioxide is not able to get in. Instead, we're going to go over the other pathway here. So let me draw you a diagram of what goes on here. So this is going to be our plant. Let me make it green here. This is going to be our leaf. And I'm going to draw a stomata in here for us. Maybe we'll draw two. Why not? Okay. So there's our stomata. Now, underneath, we have our epidermis cells, which we talked about. We are also going to have our mesophyll cells. I'll draw a few in here. I'm not going to go crazy with them. Okay. All right. So these are our mesophyll cells. Right. Now, if the stomata is open, so here's our stomata, the CO2 can readily just diffuse into the cell. But along with having these mesophyll cells, we also have different cells underneath that also can go through photosynthesis. And they are called bundle sheath cells. Okay. Here's the issue. Our mesophyll cells are going to take the carbon dioxide. The carbon dioxide is not going to be able to get to our bundle sheath cells. Ruh-roh, right? We don't want the bundle sheath cells to not have any carbon dioxide, because then they're not going to be able to make our glucose. So here's what happens. In our mesophyll cells, we're going to bring in the carbon dioxide. Bring in our carbon dioxide here. All right. Instead of combining with RUBP, it's going to combine, that's an E, sorry. It's going to combine with something called PEP. All right. It's called phosphoenol pyruvate. Okay. And what that's going to do is it's a 3-carbon molecule. So 1, 2, 3. What's going to happen here is it's going to combine with our carbon dioxide to form a 4-carbon molecule. So when these two combine, we are going to get 1, 2, 3, 4 4-carbon molecule called oxaloacetate. Okay. How this occurs is right here we have an enzyme called PEP carboxylase. Now the thing about PEP carboxylase is it fixes carbon but not oxygen. And that's fantastic for our C4 plants because we don't have to worry about the oxygen levels going up and up and up because this PEP carboxylase enzyme is never going to bind our oxygen. It does not have an affinity for oxygen. All right. What's going to happen after this? And there's stuff around the side, guys. Like I said, oxaloacetate, it's unstable. We are going to make it into a different 4-carbon molecule called malate. Okay. And then what's going to happen is the malate is then going to go back to our PEP, and it's going to release some carbon dioxide, okay? And that carbon dioxide will go to other cells. All right. Now, what's going to happen here, if we got this, I'm going to erase the top here. What's going to happen is I showed you the cycle, but I did not show you where the cycle happens. So if we have up here, this is going to be our mesophyll cell. Mesophyll cell. Oops. And down here, we're going to have our bundle sheath cell. Okay. Connecting these two, going to have a little passageway. Remember we talked about plasmodesmata and things can be transferred? So in our mesophyll cell, we're going to go from our PEP to our oxaloacetate to our malate. The malate is going to go to the bundle sheath cell. And when the malate is in our bundle sheath cell, that right there is when we're going to give off the carbon dioxide, and then it's going to go back to PEP, and the PEP is then going to go back up to our mesophyll cell. Okay? So that is what's going to happen here in order for the bundle sheath cell to get the carbon dioxide so then it can go through our Calvin cycle. All right? And that is the C4 pathway, guys. All right? So the malate's going to go to our bundle sheath cell, give off carbon dioxide, go back to PEP. PEP's going to go back up, and then that process will continue in order to supply the bundle sheath cells with our carbon dioxide. And that's it for C4 pathways. So again, these are going to be things like corn, sugarcane, crabgrass, all right? Those types of plants are going to go through the C4 pathway. Oh, our lovely pineapples too. We didn't talk about those types of plants yet, right? We're going to have another pathway. So we had the sugarcane, which is going to do C4 pathway. We're going to have the pineapple plant, which I'm not going to go crazy with the CAM pathway, guys, and drawing it and everything. It's going to be pretty much almost identical to our C4 pathway. The difference here is that when we have our C4 pathway, our Kelvin cycle, yeah, anytime it can occur. With CAM pathways, these are going to be our desert plants. So they do not want their stomata open during the day. If their stomata is opening during the day, that means our water is going to diffuse out of the plant, meaning, hey, no photosystem 2 anymore because we don't have water to fuel photosystem 2. We can have as much light as we want, but no water, no photosystems. So what's going to happen here for our CAM pathways is the plants are going to open their stomata at night, bring all their carbon dioxide in at night. I'm going to give you a quick diagram here, nothing crazy like I said, guys. It's going to be quick. So I'm just going to say stomata open here. Okay. And again, this would be at night because it would be closed during the day to make sure that water stays in. Let me draw my mesophyll cell here. There's my mesophyll cell. Okay, at night, we are going to bring in our carbon dioxide. It's going to come in, all right? Okay. It's going to combine with our PEP. We're going to form our malate. And then we're going to go back to PEP later on, okay? And that's going to, just like last time, give off carbon dioxide, okay? So what happens here is we are going to store the malate after we form it. We're just going to store it so that during the day, when the photosystems are occurring, the malate during the day can then be used, go to the PEP, release the carbon dioxide, so that when we have the ATP, when we have the NADPH ready from the photosystems, now we can fire that Kelvin cycle during the day. Oh, sorry, guys.
Mr. Yoder, please dial 5404, Mr. Yoder.
So whenever the, like I said, guys, whenever we have the ATP, whenever we have the NADPH available for us from the photosystems, we are then during the day going to use that malate, form it into our carbon dioxide, release that, and then go back to PEP. We do not care what happens with PEP after this. That's not, we don't care. We're going to use the carbon dioxide to then do the Kelvin cycle during the day. So what's the goal of these CAM plants? The goal is to take in CO2 at night to be used during the day. All right, that is our goal of the CAM plants, so that the Kelvin cycle can take place. All right. So there is the CAM pathway. Right. So this is just a review of everything with photosynthesis, guys. If the plant makes enough sugar, or it has an excess, doesn't need any more energy, it's going to store it as starch. So that way, if it needs it, we got this nice big polymer of starch. We could start plucking off monomers of glucose, of sugar, transfer them over to the mitochondria. The mitochondria can take them in and then make them to fuel our ATP. Okay, so that is the end of our lovely whole chapter. We went through a bunch of different Kelvin types of plants and how they fuel the Kelvin cycle there. But that's the end of our photosynthesis chapter, guys. We went through a lot. Take time to learn all of your diagrams. Make sure you know what comes out of each photosystem. Make sure you know what goes into each photosystem. Make sure you know what goes into the Kelvin cycle and what comes out of it, and how that Kelvin cycle continues to go. So it's a cycle. All right, guys, that is the end of the chapter on photosynthesis. Have a good rest of your day.